morning. It's September 17th, and this is your daily brief in science. Here's everything you need to know. Researchers from Waseda University have utilized artificial intelligence to analyze facial expressions among Japanese university students, focusing on subtle cues linked to subthreshold depression. These students with mild depressive symptoms tend to display less expressive, friendly, natural, and likable faces, yet their ability to judge others' faces remains unaffected. The study identified 6 facial muscle movements, including eyebrow raising, eyelid lifting, lip stretching, and jaw dropping, which were more prevalent in students with mild depression. 5 of these movements showed a correlation with depression severity scores. The findings suggest that subthreshold depression is associated with muted positive expressions and specific eye and mouth movements, but it does not significantly impact first impressions or social perceptions. While facial analysis tools show promise for early detection, limitations such as cultural specificity, small sample size, and reliance on self-reported data highlight the need for further validation across diverse populations. The research indicates that early depression may share biological links with fear responses, and reduced positivity in facial expressions could serve as an early indicator of at-risk individuals. This study marks progress in digital mental health screening by providing a scalable, non-invasive method to monitor mental health, especially as student mental health issues increase worldwide. The authors suggest that AI-based facial analysis could enable early detection of depression before clinical symptoms appear, allowing for timely intervention in educational and workplace settings. However, experts emphasize that AI tools should complement traditional assessments and stress the importance of further validation across different populations. Ethical considerations, including privacy, consent, and cultural limitations, are crucial in deploying AI for mental health diagnostics responsibly. Researchers at Monash University have developed a new carbon-based material called multi-scale reduced graphene oxide, or MRGO, which significantly enhances the energy storage and power delivery capabilities of supercapacitors. This material is produced through a rapid thermal annealing process that creates a highly curved graphene structure with precise ion pathways. These pathways enable supercapacitors to charge quickly and store high amounts of energy. Supercapacitors made with MRGO have achieved volumetric energy densities up to 99.5 watt-hours per liter and power densities of 69.2 kilowatts per liter. They also demonstrate long-term stability and scalability, with performance metrics among the best reported for carbon-based devices. The technology offers excellent cycle stability and fast charging performance, making it suitable for applications such as electrified transportation, grid stabilization, and consumer electronics. The production process is scalable and compatible with Australian raw materials, opening pathways for potential commercial manufacturing. Experts believe these rapid-charging supercapacitors could replace batteries in various sectors due to their ability to charge quickly and deliver high power. The research has been published in Nature Communications, underscoring its significance in the field. Ionic Industries, a Monash spin-out founded in 2014, is actively working to commercialize this graphene technology and is already producing quantities for market applications. Dr. Philip Aitchison, CTO of Ionic Industries, stated that partnerships are underway to bring this high-performance energy storage technology to market. A recent study sheds light on how the immune environment within lymph nodes influences the spread of aggressive breast cancers, such as triple-negative breast cancer. Researchers found that tumor cells manipulate sentinel lymph nodes before metastasis occurs, creating a pre-metastatic niche that suppresses immune responses and facilitates cancer spread. Clinical analyses of patient lymph node samples confirmed immune and stromal changes similar to those observed in preclinical models, emphasizing the pathway's relevance in human cases. The research demonstrates that tumor cells exploit innate immune mechanisms, particularly through toll-like receptor 4, or TLR4, activation in fibroblastic reticulum cells, or FRCs. This activation leads FRCs to release cytokines like CCL2 and CCL7, attracting monocytes that become immune suppressive and inhibit T-cell activity, aiding tumor immune evasion. Blocking TLR4 in mouse models, especially when combined with PD-1 immunotherapy, significantly reduces lung metastases in TNBC. Led by Dr. Angela Rydell at Würzburg University Hospital, the study highlights how TNBC leverages lymph node environments to escape immune defenses and promote metastasis. The findings reveal that immune-suppressive niches within lymph nodes, characterized by clusters of suppressive monocytes and FRCs, support tumor cell survival and spread. Advanced techniques like spatial transcriptomics and single-cell RNA sequencing identified high PDL1 expression on monocytes, which could serve as a biomarker for immunotherapy responsiveness. Overall, this research underscores the importance of targeting immune evasion mechanisms early in cancer progression. It also emphasizes that integrating immune insights into therapy, including lifestyle factors like diet and exercise, may improve outcomes by supporting immune health. The work is supported by significant funding from organizations such as the German Cancer Aid and MRF, reflecting its potential to influence future cancer treatments. A new study published in Nature Communications highlights promising progress in stroke recovery research. Researchers transplanted reprogrammed human blood-derived neural stem cells into mice 1 week after an ischemic stroke. The results showed significant recovery, including reduced inflammation and increased growth of neurons and blood vessels. The transplanted cells mainly differentiated into GABergic neurons, which are linked to stroke recovery, suggesting the brain environment influences stem cell development. This therapy also helped preserve the blood-brain barrier, reducing brain edema and infiltration of harmful substances. Advanced AI behavioral analysis demonstrated that treated mice regained complex motor skills, such as navigating uneven surfaces and improved gait. These improvements indicate both cellular regeneration and functional motor recovery. The study identified key signaling pathways activated by the transplanted cells, related to neuron regeneration and connectivity, which could be targeted by existing drugs. Further research is exploring how to enhance these pathways and assess the long-term effects of stem cell transplants. Long-term studies aim to evaluate the persistence and integration of transplanted cells to support ongoing recovery. Delivery methods like endovascular injection are being tested to make treatments more practical for humans, with some clinical trials already underway for Parkinson's disease in Japan. Additional safety measures, including control systems to prevent uncontrolled growth, are also in development. Researchers continue to explore ways to extend treatment windows beyond current limits and improve the safety and efficacy of stem cell therapies for stroke patients. Researchers at Shibara Institute of Technology, led by Professor Naomi Osakabe, have used the latest AI model, AlphaFold 3, to predict the detailed 3D structures of all 25 human bitter taste receptors, known as T2Rs. This achievement marks a significant step forward in receptor structural biology, overcoming previous challenges in obtaining accurate structural data. The study compared AlphaFold 3 predictions with earlier AlphaFold 2 results and experimental structures, finding AlphaFold 3 provided more precise models, especially for T2R14 and T2R46, which were previously characterized only through cryo-electron microscopy. AlphaFold 3 consistently outperformed previous models in accuracy, addressing past limitations in resolving T2R structures. T2Rs are expressed not only in the oral cavity, but also in gastrointestinal neuropod cells, where they influence gut-brain communication, glucose regulation, and appetite control. These functions have implications for metabolic diseases such as diabetes. The research highlights the importance of T2Rs beyond taste perception, particularly in the gut, where they help regulate glucose homeostasis and appetite. Structural modeling of T2Rs is crucial for understanding their functions and could guide future studies on taste variability and receptor-ligand interactions. Analysis revealed conserved intracellular regions responsible for signal transduction and highly variable extracellular regions involved in ligand recognition, explaining their broad specificity. Researchers classified T2Rs into 3 clusters based on structural similarities and differences, aiding in understanding their specific responses to bitter compounds. Predictions indicate that diverse bitter molecules bind within the extracellular pockets of T2Rs, activating G proteins like gusteucin to trigger intracellular signaling pathways. Published in July 2025, this study exemplifies how integrating AI with molecular biology enhances understanding of taste receptors and their broader physiological roles, with potential health applications. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.